So welcome my students. In previous class we have studied the first artificial recombinant DNA. How the world's first recombinant DNA synthesized by Stanley Cohen and Herbert Boyer. That we have seen. Okay. In today's class, let us study the further things. The basic steps of genetic engineering. The genetic engineering involves some basic steps. They are as follows. First one, identification of DNA with desirable genes. Second, introduction of the identified DNA into the host. And third, maintenance of introduced DNA in the host and transfer of the DNA into the progeny. These are the three steps. Identification of DNA with the desirable genes. So this is very, very important. It is the first and the basic step of genetic engineering. Identification of DNA with desirable genes means first you must identify the DNA. Say for example, I want to introduce in plants a disease-resistant gene, or I want to introduce a drought-resistant gene, or I want to introduce a high-yielding gene. Or in case of animals, for example, in the improvement of milk in cows or buffaloes, the milk producing gene we must first identify. Similarly, the disease resistant gene of the crop we must identify, or the drought resistant gene must identify. Say for example, ನೀವು ಹಳ್ಳಿಯಿಂದ ಬಂದರೆ ಗೊತ್ತಾಗಿರುತ್ತೆ, ಬಾಜ್ರಾ crop is there. Bajra means sajje. That bajra, it is having natural drought resistant character. ಮಳೆ ಇಲ್ಲದಿದ್ರೂ ಬೆಳೆ ಹಾಗೆ ಇರುತ್ತೆ ಹೊಲದಲ್ಲಿ. ಮಳೆ ಬಂದಾಗ ಚೆನ್ನಾಗಿ ಬೆಳೆ ಬರುತ್ತೆ. It can overcome the drought condition. How it is possible means because of the gene. Drought resistant gene is there in that plant bajra. ಇನ್ನೂ ನಿಮ್ಗೆ ಹೇಳಬೇಕಂದ್ರೆ ಕೆಲವು ಸಾರಿ ಬೇಕಾದ್ರೆ ನೆಗಡೆ ಆಗಿರುತ್ತೆ. ನನ್ನ ಫ್ರೆಂಡ್ಗೆ ಪಕ್ಕದಲ್ಲಿ ಕೂತ್ಕೊಂಡ್ರೆ ಕೆಲವರಿಗೆ ನೆಗಡೆ ಆಗುತ್ತೆ ಇಮ್ಮಿಡಿಯೇಟಾಗಿ, ಇನ್ನು ಕೆಲವರಿಗೆ ನೆಗಡೆ ಆಗೋದಿಲ್ಲ. ಬಿಕಾಸ್ ಈಸ್ ಹ್ಯಾವಿಂಗ್ immunity, against that virus is resistant. ಅದನ್ನೇ ನಾವೀಗ ಕೋವಿಡ್ನಲ್ಲಿ ಹೇಳ್ತಕ್ಕಂಥದ್ದು. ಈವನ್ ಡಿಸೀಸ್ ಸಸೆಪ್ಟಬಲ್ ಇದ್ದರೆ ಬೇಗ ಇನ್ಫೆಕ್ಷನ್ ಆಗುತ್ತೆ. ರೆಸಿಸ್ಟೆಂಟ್ ಇದ್ದರೆ, ವಾಟ್ ವಿ ಕಾಲ್ ಇಮ್ಯುನಿಟಿ ನಮ್ಮಲ್ಲಿ ಇದ್ದರೆ, ಇನ್ಫೆಕ್ಷನ್ಸ್ ಬಂದ್ರೂ ಏನೂ ಆಗಲ್ಲ ಅಂತ ಹೇಳ್ತಿರ್ತೀವಿ ನಾವು. ದಿಸ್ ಈಸ್ that immunity is in our body. It is because of the gene. That gene we must first identify. Say for example, disease resistant gene, high yield gene, or drought resistant gene in the crop we must identify first. That is the gene, as you know, you have studied molecular basis of inheritance. The genes are always found or located in the DNA molecule. It means we have to identify the DNA which must contain the desirable genes. Okay. Next, once we identify the DNA with the desirable gene means introduction of the identified DNA into the host. Now, in the second basic step of RDNA technology or genetic engineering, the identified gene, what we call desirable gene, it is also called gene of interest. It is also called alien gene, also known as foreign gene. This gene we must isolate from the DNA molecule. How to isolate the DNA from the DNA molecule, we will discuss later in detail. But now, in a brief manner, if the DNA is treated with a special type of enzymes called restriction endonucleases that cut the DNA molecule, and we may obtain the desirable gene. That gene should be introduced into the vector. You will come to know what is vector later on. Vector means it is a vehicle which transfers the gene. We call cloning vehicle. Okay. The identified gene, that is foreign gene, is transferred to, for example, plasmid is a vector, into plasmid. Then that plasmid must be introduced into the host plant. Host may be bacterium also, or any crop plant. That is the second step. And the third step is maintenance of introduced DNA in the host and transfer the DNA into the progeny. So once we introduce the gene into the host means we must maintain that one, then you must multiply that gene or multiply the progenies. ಒಂದು ಸತ್ಯ ನಾವು ಜೀನನ್ನು ಹಾಕಿದ ಮೇಲೆ ಹೋಸ್ಟ್ ಸೆಲ್ಲಲ್ಲಿ ಅವನ್ನು ನಾವು ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಬೇಕು. Actually, we call it as cloning in case of bacteria, say. So then we get product from that progeny. From the desired gene, we get the product. So these three are the basic steps of genetic engineering, very important for 2 or 3 marks. Mention the basic steps of genetic engineering. Okay, so that's all. Now we shall pass on to the tools of genetic engineering. The genetic engineering is also called recombinant DNA technology. We have already discussed the definition of genetic engineering. In genetic engineering work, the scientist is going to engineer the gene. So, that technology we call RDNA technology. ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ನಲ್ಲಿ ನಾವು ಜೀನನ್ನು ಹಾಕ್ತೀವಿ ಹೋಸ್ಟ್ ಆರ್ಗ್ಯಾನಿಸಮ್ನಲ್ಲಿ. ಹೋಸ್ಟ್ನಲ್ಲಿ ಡಿಸೈರ್ಡ್ ಜೀನ್, ಬೇಕಾದಂಥ ಜೀನನ್ನು ನಾವು ಹಾಕ್ತೀವಿ. ದೆನ್ ಅದರಿಂದ ಆ ಜೀನಿದ್ದ ಪ್ರಾಡಕ್ಟನ್ನು ನಾವು ತಗೊಳ್ತೀವಿ, ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಓಕೆ, ನಾವು ಜೀನ್ಸನ್ನು ಹಾಕ್ತೀವಿ ಅಂತ ಅಂದರೆ ಸುಮ್ಮನೆ ಹಾಗೆ ಹಾಕಲಿಕ್ಕೆ ಬರೋದಿಲ್ಲ. ಸೇ ನಾವೇನಾದ್ರೂ ಒಂದು ಕಾರ್ಪೆಂಟ್ರಿ ವರ್ಕ್ ಮಾಡ್ತಾ ಇದ್ವಿ ಅಂತಂದರೆ ಇನ್ಸ್ಟ್ರೂಮೆಂಟ್ಸ್ಗಳು ಬೇಕು. ಅವು ವಿತೌಟ್ ಇನ್ಸ್ಟ್ರೂಮೆಂಟ್ ಒನ್ ಕಾಂಟ್ manufacture or prepare the chair, you can say now, or a door. Tools very necessary. Like that, the tools of genetic engineering. It means in genetic engineering or in RDNA technology, some tools are necessary. What are the tools? Say, one is host. Normally, E. coli cell is used as host. Need not be always E. coli. E. coli host. Second, vectors. Vectors usually, commonly, plasmid are used as vector, but not always. Vectors also, other vectors are also there. Then restriction enzymes. They, in brief, we call REN. Enzymes necessary which cut the DNA molecule actually. Then polymerase enzymes. These enzymes construct the DNA molecule. Ligase enzymes, these join two DNA fragments. That's why we call a joining enzyme. Next tool is bioreactor for multiplication and to produce the progeny, then to produce the product. Okay. So host E. coli, Escherichia coli, is commonly used as host in genetic engineering. Okay? Now I hope you have understood the basic steps of genetic engineering. The detailed steps of genetic engineering you are going to study later on. Okay. So now we shall pass on to our next one, that is the host. So detailed study of these things, tools. Host, it's not discussed in your textbook, but host, commonly used host in genetic engineering is E. coli. E. coli is a bacterium which is living in the colon region of human intestine. Okay. This E. coli bacterium having cell wall, then flagella, next to the cell wall cell membrane is there, then cytoplasm is there. In the cytoplasm bacterial DNA is there. In addition to that, it is also having plasmid. So that's all about the brief account of E. coli bacterium. Okay. So first point. So host cells. ನಾವು ನೋಡಿದ್ದೀವಿ ಈಗ. Next, very important, restriction enzymes. ಸ್ವಲ್ಪ ಸ್ಪೆಲ್ಲಿಂಗ್ ಮಿಸ್ಟೇಕ್ ಆಗಿದೆ ಇಲ್ಲಿ. Restriction enzymes. Restriction enzymes, REN we call in brief. Restriction endonucleases. Okay. The first restriction endonuclease enzyme, REN, was Hind second. Actually, capital ಆಗಬೇಕಾಯಿತು. Hind second. Discovered by Hamilton Smith in the year 1970 in a bacterium Haemophilus influenzae. Okay. Now, why we call restriction endonuclease means these enzymes are always, their action is restricted on the DNA molecule. I will discuss later on that also. Restriction endonuclease enzymes always cut the DNA molecule at a particular point or specific point on DNA molecule by identifying 6 base pairs on DNA. These 6 base pairs we call recognition sequence. Enzyme ಯಾವ ಥರ ಆ್ಯಕ್ಟ್ ಮಾಡುತ್ತೆ ಅಂತಂದರೆ, the enzyme first ಐಡೆಂಟಿಫೈ ಮಾಡುತ್ತೆ recognition sequence. Okay. Then what is this recognition sequence ಅಂದರೆ? The specific base sequence. Specific means particular base sequence. Say for example, GAA-TTC, specific nucleotides on DNA is known as the recognition sequence. Okay? All enzymes identify the recognition sequence. Okay. Now, why these enzymes are called restriction endonuclease means Because these enzymes cut the DNA molecule only at particular point or at particular site. Hence, called restriction endonucleases. ಈ ಎನ್ಸೈಮ್ಸು ಡಿ ಎನ್ ಎನ ಮಧ್ಯದಲ್ಲಿ ಒಂದು ನಿಗದಿತ ಜಾಗದಲ್ಲಿ ಕಟ್ ಮಾಡ್ತವೆ. ದಟ್ ಈಸ್ ವೈ ದ ನೇಮ್ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯೇಸ್. ಓಕೆ, ನೌ these enzymes are commonly known as molecular scissors or molecular knives because these enzymes cut the molecule. That is, the enzymes cut the DNA molecule, hence called restriction endonucleases. Okay. Scientists discovered more than 900 restriction endonucleases enzymes. What's the number? 900 different types of enzymes are discovered by the scientists. And these enzymes are isolated, means obtained, you may take. Obtained from 230 different types of bacterial strains. How many number? 900. The bacterial strains? 230. Very important for NEET examination. Okay. What are molecular scissors? For 1 mark. For the main examination. What is recognition sequence? For 1 mark. For the main examination and CET or NEET. Scientists discover 900 enzymes and from 230 different types of bacteria. Look here now. Each recognize different recognition sequence. I will give you some enzymes. Examples of some enzymes. Restriction endonucleases. Eco or first. Eco or second. Alu first. Hind second. Hind third. Etc. Eco or first means Escherichia coli. From the bacterium we obtained. Alu first means the enzyme obtained from Arthrobacter luteus. The enzyme. Sorry, the bacterium. Hind second, the enzyme obtained from influenza. What's that? Haemophilus influenzae. Okay. The enzyme name is given after the bacterium obtained. Okay. So that's so. Next we shall see. Naming of restriction endonuclease enzymes for 3 marks. Write note on. Naming, say for example, eco or first, eco or second, alu first, hind second, hind third, like that. How the naming is done? The naming is done on certain principles. Say for example, eco or first, basally nodi, ಕೆಳಗಡೆ ನೋಡಿ, eco or second, etc. ಇದೆ example. In the ecor first, the first letter E always refers to the genus name. That is, ecor first obtained from Escherichia, the bacterium. The name of the bacterium from which isolated. Now you see, co is there. Next two letters, that is, example, co refers to the species name. It is written in italics. You know, italicized words ಡಿಕ್ಷನರಿನಲ್ಲಿ ನೋಡಿದರೆ ನಿಮಗೆ ಗೊತ್ತಾಗುತ್ತೆ. ಕೆಲವು ಸಾರಿ ರಾಮ ಈಸ್ ಎ ಬಾಯ್, ರಾಮ ನೌನ್ ಅನ್ನೋದು ಐಡೆಂಟಿಫೈ ಮಾಡೋದಕ್ಕೆ ತುಂಬ ಸಣ್ಣ ಅಕ್ಷರದಲ್ಲಿ ಬರೆದಿರ್ತಾರೆ. ಇಟಾಲಿಸೈಜ್ಡ್ ವರ್ಡ್ ಅಂತ ಕರಿತೀವಿ. ಲೈಕ್ ದಟ್. ದ ನೆಕ್ಸ್ಟ್ ಥರ್ಡ್ ಲೆಟರ್ ಆರ್ indicate the strain of bacterium. Then one more letter is also there. You see carefully. For example, ecor first is there, ecor second is also there. Okay. The Roman letter first, second, third indicates the number of endonucleases isolated from the bacterium. Okay. For example, ecor first isolated enzyme. Ecor second isolated enzyme. Second time. So that's all about the naming of restriction endonucleases. Very important for the main examination. Next we shall see how these enzymes function. Functioning of enzyme is also very, very important. Why we call them restriction enzymes? Restriction enzymes belonging to a larger class of enzymes called nucleases. Nucleases are classified into two types: exonucleases and endonucleases. What are exonucleases? Exonucleases are a kind of nuclease enzymes which cut or remove the nucleotides from the ends of DNA. DNA ತುದಿನಲ್ಲಿ ಕಟ್ ಮಾಡ್ತದೆ. Sticky ends are not formed. Okay. Next, another type of enzymes are there called endonucleases.